Cancer. Dr. David Breslow is an assistant professor in the Department of Molecular, Cellular, and Developmental Biology. He received his PhD from the University of California at San Francisco, and among other things has been studying the biology of cilia to improve our understanding of cancer, and look forward to learning more about that. So thank you.
All right, thank you. All right, so yes, I'm an assistant professor in the MCDB department, so my lab is up on Science Hill. And our main research focus is on understanding the functions of this cellular structure, which is the primary cilium. And I should say at the outset that I'm not a dyed-in-the-wool cancer biologist, but we're focused on the basic biology of cilia. But I think, first of all, that biology is interesting, and I'm happy to share some of it with you today. And also, there's some exciting connections emerging that we're eager to pursue of cilia to cancer. So first of all, what is the primary cilium? So as you can see in these EM and light microscopy images, the primary cilium is a microtubule-based protrusion from the cell surface. It's actually quite evolutionarily conserved, being likely found in the last eukaryotic common ancestor. And unlike other types of cilia seen in other organisms or in specialized tissues in the body, the primary cilium is distinct because there's only one primary cilium per cell, and it's not motile. However, it's found actually on most cells in the human body. So for a long time, it was unclear what functions primary cilia have, if any, but what's generated a lot of excitement in the last 10, 15 years is the realization that the cilium is actually a key organizing center for a variety of different signaling pathways in the cell, acting analogously sort of like as an antenna of the cell. One of the ones I'll focus on is in hedgehog signaling, but the number of different other forms of signaling through other receptors and ion channels are organized by cilia. And because of these important roles of cilia in signaling, it's maybe not surprising that mutations that disrupt cilium function lead to a variety of different developmental disorders, and also that aberrant signaling through cilia is implicated in several cancer types, as I'll describe in more detail. So if we look at a little bit more closer detail at the structure of the cilium, there's a few features to highlight. So as I mentioned, it's a microtubule-based structure, and at the core of it are these so-called axonemal microtubules that extend from the base of the cilium, which is formed by the mother centriole, which basically docks at the cell surface here and gives rise to these axonemal microtubules. And this microtubule core is surrounded by the ciliary membrane, which is really essentially a subdomain or protrusion from the plasma membrane surface of the cell. And actually, it's even so, the main point here is that this is actually a fairly complex and intricate structure, even though it's quite small. So in terms of the scale, it's about 1 to 6 microns in length and about 300 nanometers in diameter. And what we've realized in the last several years is that even though the cilium doesn't have a membrane surrounding it, it has a distinct complement of both membrane and soluble proteins that support its function and that are highly concentrated within the cilium. Many of these proteins are actually have important roles in signaling, so they include membrane receptors and transducers that help support the signaling activities of cilia. In the context of disease, there are a number of congenital genetic disorders that have now been identified that first went by a variety of different names, but now we know that what they share in common is that these disorders are essentially can be known as ciliopathies because they are due to inherited defects in cilium function. The symptoms associated with ciliopathies are quite diverse and actually were a puzzle for a long time to clinicians trying to understand how, for example, you can get retinal degeneration, anosmia, a variety of craniofacial and other skeletal defects, kidney cysts, obesity, cognitive impairment, all resulting from a single mutation. And now we understand that this occurs because these many different symptoms reflect the many different tissues and cell types in which primary cilia and ciliary signaling are important. Thanks to some really great progress from the human genetics research side, there's now many different causative genes that have been identified for ciliopathies. Actually, about over 90 such genes are now known, although a key challenge in the field still remains actually understanding what exactly these gene products do at a molecular level, and also there's still unidentified disease genes that likely explain the cause of disease for patients where these mutations are still unknown. These disorders reflect what happens when ciliary defects are basically inherited congenitally. I think an exciting area that is beginning to gain some traction is understanding what the functions of cilia are in adult tissue homeostasis or in degenerative diseases. And there's some emerging links between ciliary defects and neurodegeneration, and also, as I'll of course describe in more detail, in cancer. So an important paradigm for understanding the roles of cilia is exemplified by the hedgehog pathway. So some of you may be familiar with the hedgehog pathway for its important roles in organizing embryonic patterning and also in tissue homeostasis. And thus aberrant hedgehog signaling can lead to developmental disorders, but also can give rise to different forms of cancer. And in particular, in a subset of medulloblastomas and basal cell carcinomas, hedgehog signaling is basically sufficient to drive the disease. And this has generated a lot of interest in means to therapeutically target the hedgehog pathway for these cancers. So here's a patient actually who has a metastatic form of medulloblastoma who was treated with a Genentech developed compound that inhibits the hedgehog pathway. And this produced a quite nice response in this patient. Unfortunately, however, subsequently there was a recurrence of disease in this patient who subsequently died. And so I think this illustrates both the promise of targeting the hedgehog pathway in cancer therapy, but also the challenges in that we still need to learn more about hedgehog signaling to improve the therapeutic opportunities. One of the things that we still need to learn about is the role for cilia in hedgehog signaling. And something that generated really a lot of surprise in the hedgehog signaling field was the discovery about 10, 15 years ago now that the cilium actually provides the essential environment in which hedgehog signaling occurs. And so now we know that all of the key components of the hedgehog pathway, including this protein smoothen, which was actually the target of this drug, localizes to these structures here, which are the primary cilia. And we also know that any defect in primary cilium assembly or function will compromise hedgehog signaling. And basically, cilia are required for hedgehog signaling in vertebrates. So how does this relate to the roles of cilia in cancer? So one thing I want to emphasize is that the roles of cilia in cancer are really context specific. In some cases, cilia are actually required for tumorigenesis. And this is exemplified by these hedgehog-driven cancers in medulloblastoma and basal cell carcinoma, where cilia are needed for tumorigenesis, and then therefore disrupting cilia or ciliary signaling is potentially beneficial. And a nice piece of data kind of demonstrating this principle is in this mouse model of medulloblastoma in which hedgehog pathway signaling is activated. And when you combine this hedgehog activating mutation with a ciliary mutation that disrupts cilium function, that greatly extends the survival in this mouse model of medulloblastoma. So to better understand the roles of cilia and hedgehog signaling, we recently conducted a genome-wide functional screen to better understand these processes. And really for this screen, we had a few different goals. One of our goals was to better understand the hedgehog pathway. There's actually a number of outstanding questions in exactly how hedgehog signals are transduced intracellularly. And also to better understand the biology of cilia, to understand unanswered questions, for example, in how cilia are formed and how they function, and particularly a major unknown is how is it and why is it that they provide the essential location in the cell where hedgehog signaling happens. And more broadly, I think we had in mind that this type of screen would give us some new insights into the roles of cilia in disease, both to help maybe identify new ciliopathy disease genes and also to identify genes that might be candidate therapeutic targets for these hedgehog-driven tumors. And so to carry out the screen, I'm going to skip over many of the details. Most of it they are published here. We needed two key reagents to carry out the screen. One is we established a cell line in which we had a hedgehog reporter gene that reads out the transcriptional activity through the hedgehog pathway and converts that into expression of this reporter gene, which confers resistance to the antibiotic blastocidin. So that cells with high levels of signaling become resistant to blastocidin, and cells with low levels of signaling, due perhaps to mutation, have low levels of resistance to blastocidin. So basically, we can convert differences in cilia function and hedgehog signaling into differences in viability in the presence of blastocidin. And then we combined that with a genome-wide CRISPR library in which each gene in the genome is targeted by 10 different guide RNAs. And so with this, we can mutate every gene in the genome and read out which genes affected cilia and hedgehog signaling through their effects on this blastocidin reporter and their cell viability in the presence of blastocidin. So what did we find in this screen? So we were actually very pleasantly surprised that the screen seemed to work quite well. And we could tell that it worked well in two different ways. One, as you can sort of see from this wide spectrum of hits, is that the hit genes we identified in the screen really broadly covered different aspects of cilia biology. And basically, these genes correspond to virtually every known protein complex and ciliary substructure that is known to be required for the proper function of cilia. So the screen really broadly covered different aspects of cilia biology, which was one of our goals. The other way we could tell the screen was successful is that for some of these categories of genes, we basically know all of the genes that comprise that category, and they all should be important for cilia. So we could ask what fraction of these genes did we successfully identify as hits? And in many of these cases, we found basically all or nearly all of the genes in these categories as hits in our screen, suggesting that we really were approaching saturation in our screen, and there were probably few false negatives that escaped our screen. So we've then followed up on a number of new genes that were previously uncharacterized and newly implicated by our screen in cilia biology to try to understand their cellular functions. And the types of lessons that we could learn from characterizing some of these new genes, I'll spend just a few minutes describing. So one thing we learned is that this type of information can be very valuable for understanding ciliopathy genetics. And this is exemplified by the gene TXNDC15. And we came across a human genetics study on a ciliopathy patient pedigree where they did exome sequencing on these individuals to try to identify the causative mutation. And as is often the case in exome sequencing of a small pedigree, it's difficult to narrow down to a single definite causative gene. Often one is left with a few candidate genes, and it's difficult to say which of the candidates is really the causative gene or not. And so that was the case in this particular pedigree. There were 3 genes that fit all the criteria, and the authors of this study favored this gene XOC4 as likely the causative gene because of some prior work linking members of this gene family to cilia. But we were interested to look through our screen results and see that XOC4 and ETV1 were not hits in our screen, but this gene TXNDC15 was a strong hit in our screen. So this is a transmembrane protein with a thyroredoxin domain, and the geneticists identified a particular mutation loss of these 5 amino acids in this domain. So we thought maybe we can actually explore this further even into cell culture-based models. So we did a hedgehog signaling assay in cell culture, where basically we take cells and either leave them unstimulated or stimulate them with hedgehog ligand and look at the level of signaling response. So you can see in this gray bar, when we have cells with a control guide RNA, basically wild type cells, they produce a pretty robust response in turning on a reporter. And cells with either of 2 different guide RNAs targeting TXNDC15 have strongly reduced levels of hedgehog signaling response. We can rescue this defect in signaling by reintroducing a wild type copy of the TXNDC15 cDNA. But what was more interesting is when we tried to repeat this rescue experiment with the mutant allele that had been identified in these patients. So we recreated this 5 amino acid mutation loss. And when we did this rescue experiment, we found basically there's no rescue at all with the mutant form of TXNDC15. The level of signaling is the same as if we just have GFP alone. So together, these findings told us that TXNDC15, first of all, was required for hedgehog signaling, and second, that the patient mutation appears to be pathogenic and non-functional. And so this was quite interesting to us, and we got back in touch with actually the human geneticists who first carried out this study, and they actually told us they had just found another patient who had a mutation in TXNDC15. So there was a really nice convergence between our cell-based studies and their ongoing human genetics efforts. And so I think more broadly, though, I think this provides a strategy for functional testing of candidate genes that are emerging from human genetics, because this is actually a quite common scenario that there might be multiple candidate genes, and this provides a way to try to sort through that type of information. The other types of things we tried to learn from our screen was about the basic biology of cilia. And just one brief example of that is one hit gene in our screen. There was a small GTPase known as RAB34. And the function of RAB34 is not well characterized, but what we found is when we made knockout cells for RAB34 and we stained for the cilia, marked here in red, we basically found that while wild type cells, almost every cell has a cilium, there's basically no cilia found in RAB34 knockout cells. We also found that the protein RAB34 actually localizes to cilia, co-localizes with that protein I mentioned before, RAL13B, which is a robust marker for identifying cilia. So we can tell that RAB34 also localizes to cilia. And so in some additional work that I don't have time to discuss today, we found that RAB34 mutants fail basically to build the ciliary membrane, and the RAB34 is needed not only for cilium assembly, but also for the maintenance of cilia. But more broadly, I think our screen provides a strategy and a data set that can identify candidate therapeutic targets for hedgehog-driven tumors, because disruption of these genes clearly compromises hedgehog signaling. Okay. So that's one way in which cilia can contribute to cancer. But as I mentioned, the contributions of cilia in cancer can be context specific. So the other major context is not where cilia are required for tumorigenesis, but actually maybe the opposite relationship, which is that cilia are commonly lost during tumorigenesis. And so this has been observed now in a variety of different cancers, including melanoma, breast cancer, pancreatic cancer, and cholangiocarcinoma, as well as others. And what is found is that these tumors that normally arise in ciliated tissues lose their cilia during the tumor progression. So an example of this is in this breast cancer. So here is normal tissue where there are cilia here marked in red. And when you look at an invasive breast cancer sample, we basically see that there's no cilia present. So this raises the possibility that cilium loss is actually contributing to tumorigenesis. And furthermore, that maybe restoring cilia to these cells might actually be beneficial and might inhibit the growth of these tumor cells. And one, I think, exciting proof of principle from Brian Dinlack's lab at NYU provided some support for this hypothesis. So they looked at breast cancer cells that basically have lost the ability to ciliate to a great extent. So the level of ciliation shown here is quite low when there's a control siRNA. But when they knock down two factors that seem to restore ciliagenesis to these cells, what they found is, first of all, that this worked, and now the cells are more highly ciliated, but that furthermore, this led to a reduction in proliferative cells as seen by the decreased fraction of cells that have the proliferation marker Ki67. So in this case, restoring cilia to these cells appeared to impair their growth, which is an exciting possibility to further explore. So what exactly is going on here with the loss of cilia in these cells? How is it that cilia are lost in these cells? And I think to better understand that, we need to understand the natural conditions in which cilia are lost, which is in the context of cell cycle progression. And so what I did mention before is that the cilium is actually a quite dynamic structure. So it's typically found on cells that are in the G0 and G1 phases of the cell cycle, but that cilia are never observed on cells in mitosis. And so cilia have to be lost as cells progress through the cell cycle before they enter mitosis. Now, this actually raises a lot of questions. I think we know a fair bit about how cilia are assembled, although a lot still remains to be figured out. But we know much, much, much less about how it is that cilia are disassembled at a mechanistic level. And more broadly, I think we don't understand how cells manage to carry out cilia disassembly with such high fidelity. How are they ensuring that cilia disassembly always happens before mitosis? Furthermore, how might errors in this process contribute to the loss of cilia in cancer? And how could we maybe manipulate these pathways to restore cilia in cancer? And finally, why is it that loss of cilia in cancer seems to actually be beneficial for cancer cell growth? So what I'm going to focus on in the last minutes here is understanding this question of how is it that cells ensure cilia disassembly before mitosis? And so the way we're thinking about this is that it's like a novel type of cell cycle checkpoint. And so what is observed is that wild type cells, basically as they progress through the cell cycle, they lose their cilia and then enter mitosis. But when cilia disassembly is inhibited either genetically or pharmacologically, then this would lead to a failure to disassemble cilia and a failure to progress through the cell cycle, because this is arguably a mandatory event in cell cycle progression. And so this leads us to a model that we and others in the field are pursuing, which is that a checkpoint-like mechanism is basically monitoring and ensuring cilium disassembly in the context of cell cycle progression. So how can we test this experimentally? We have a relatively simple cell culture system where we take cells that are proliferating, we serum starve them so that they're in G0 and stop proliferating and they all form cilia. And then we add back serum growth factors to these cells. And then what this causes them to do is to disassemble their cilia and re-enter the cell cycle. And so that's in fact what we observe. So we take our cells, we add back serum growth factors, and we see that they go from being 90% ciliated to about 10% ciliated. And while this occurs, they also are progressing through the cell cycle, which we can read out by staining them for the S phase marker cyclin A. So the cells enter S phase as they disassemble their cilia. So now what happens when we inhibit cilium disassembly? So we spent actually quite a bit of time mining the literature and testing out different approaches for how to inhibit cilium disassembly, which is difficult because so little is known about how it is carried out. And what we eventually found is that low concentrations, very low concentrations of the actin poison cytokalasin D actually provide a surprisingly effective way to inhibit cilium disassembly without having broad effects on the actin cytoskeleton. So when we treat cells with cytokalasin D, what we find is that, as I mentioned, this very potently inhibits cilium disassembly, so all the cells remain ciliated even after we add back the serum growth factors. Now, what does this do to the cell cycle? we were interested to find that when we treat cells with cytoclastin D, now they are unable to progress through the cell cycle. They are unable to enter S phase, which provides evidence that this checkpoint-like mechanism is in fact operative. However, we had some concern. How do we know that these effects of cytoclastin D are really through an effect on inhibition of cilia disassembly versus some other consequence of this treatment on cells that could be unrelated to cilia? And so to test that, what we can do is repeat the same treatment that inhibits cilium disassembly, but do so in cells where we've introduced a mutation that prevents cilium formation. And so if we've prevented cilium formation, the cells should no longer care if a cilia disassembly inhibitor is present because they don't have any cilium that needs to be disassembled. So the prediction is that in this case, then, the disassembly inhibitor should have no effect on cell cycle progression. So that's what we next tested. We used mutant cells where we knocked out a gene, happens to be called SCLT1, that is required for cilium assembly. And as you can see here, these cells don't have any cilia at all. Now, what happens then when we look at cell cycle progression in the presence of cytoclastin D in these cells? What we find is that the untreated cells, as well as the cytoclastin D-treated cells, progress through the cell cycle just fine. So this provides support for our model that basically this treatment is specific for cilium disassembly because it has no effect on cells that don't have any cilia at all. So that left us fairly convinced that cells do have a cilia disassembly checkpoint, then left us with a lot of questions about how exactly it works mechanistically. And we thought maybe this is related to other types of cell cycle checkpoints that often utilize the tumor suppressor P53 to stop the cell cycle in the presence of stress or something like DNA damage. So is P53 required for this type of cilia disassembly cell cycle checkpoint? So to test this, we made P53 knockout cells and basically repeated the same experiment. So first of all, the P53 knockout cells, maybe not surprisingly, when we add back serum growth factors, they enter the cell cycle faster and they disassemble their cilia faster than wild type cells. However, what happens now when we add cytoclastin D to inhibit cilium disassembly? What we found is that cilia disassembly is still largely blocked in these cells. However, and furthermore, in the P53 knockout cells in the presence of cytoclastin D, they remain unable to progress through the cell cycle. So this tells us that the disassembly checkpoint does not require P53. So it appears in this way to be different than other types of cell cycle checkpoints. Although this difference is actually potentially interesting and maybe valuable because it means that this checkpoint could still be operative even in tumor cells in which P53 is mutated. And that maybe even in P53 deficient cells, we can exploit this checkpoint to try to inhibit their growth. Another question that we've tried to answer in the last minute here is understanding how it is that how this checkpoint works. How is it that cells sense whether the cilium has been disassembled successfully or not? And one idea we had is that maybe cells are not monitoring the presence of the cilium directly, but they're sensing indirectly whether different events are occurring at the centriole that provides the foundation, as I mentioned, on which the cilium is built. And so maybe centriolar properties are the key. So we basically, to test this, made a particular mutation in which we mutated two genes that are important for a centriole structure and asked what happens in the context of these altered centrioles. And we made two different versions of these knockout cells. And the first thing we found, which actually was completely unexpected and a surprise to me, is that just this mutation actually inhibited cilia disassembly. So now these cells appear to not disassemble their cilia the same way as the cytoclastin D-treated cells. Okay, so that was an unexpected surprise and helps us maybe get some new clues and footholds into understanding how cilia disassembly occurs. But then what happens to these cells when we look at cell cycle progression? We were quite surprised and excited to see that actually these cells with altered centrioles can progress through the cell cycle just fine, even though their cilia remain intact. And so this suggests that we found a mutation that uncouples the disassembly of cilia from cell cycle progression, and that perhaps these genes or these properties of centrioles are in fact related to how cells are sensing whether cilia have been disassembled or not. So what we're looking to understand now is what happens to these cells if we look at later stages in the cell cycle. We saw that they can enter S phase. What happens next? Do they arrest later in the cell cycle? Do they arrest in mitosis and with a cilium still present? Do they proceed through mitosis? Or does this lead to mitotic errors or some types of events that might actually contribute to cancer formation? So just to summarize, I've convinced you that cilia are important for regulation of cell signaling and cell proliferation, that they can have context-dependent roles in tumorigenesis, and that genome-wide functional screening can help to identify new regulators of cilia. And so in addition to the screen I described, we're setting up a new screen now that's targeted at understanding how cilia disassembly occurs and is regulated. Because we think that this process of cilium disassembly is an important and poorly understood checkpoint in the cell cycle that merits further study. So far, it appears that p53 is not required for this process, but that centriolar mutations do contribute to how cells operate this cilia disassembly checkpoint. And the questions that we're now beginning to focus on is what happens when the checkpoint is defective, and how and why tumors are able to suppress ciliation, and whether there are ways that we can manipulate this to restore cilia in cancer, and whether this may be beneficial. So with that, I just want to thank Max Natry, Adam Kopp, Brandon Vu, and James Chan and Sasha Hugener are at Stanford, with whom we did the genome-wide CRISPR screen. Margaret Kennedy and Anil in my lab helped follow up the genes from that screen, including TXNC15 and RAP34, and Alice and Ashik led the work on cilia disassembly and the cell cycle. Happy to take any questions.
I think there is a vast literature on the importance of centrosomes and mitotic checkpoints, and also on p53 dependent processes, ATM and CHEK2 have both been localized to centrosomes. How do the centrosome connection with cilia and with formation of mitotic spindles, what does that handle with achieve? Or how do they interact?
That's a great question. That's essentially what we would like to better understand. So first of all, a question could be asked, why do cells have this checkpoint in the first place? It's not like the DNA checkpoint where, yes, you don't want to enter mitosis before the DNA has been duplicated. That's kind of obvious. Why do cilia need to be disassembled? It may well be that, yes, the centrosomes have this other function in organizing the mitotic spindle. Now, why they can't do both things at once is unclear. There's actually work shown that cells without centrosomes can form a mitotic spindle fairly well. So if cells can make up a spindle without any centrosomes, maybe not perfectly, but pretty well, why is it so bad if the centriole would still be present? So we don't know the answer to that yet, but that's the type of question we're still investigating.
So one of those Hedgehog inhibitors actually approved for leukemia. We study it in multiple clinical trials. And what do we know about the cilia and metabolic stem cells, and how does that interact with the soil?
Yeah, that's a great point. So that's in some ways surprising, because hematopoietic cells basically all immune cells and cells derived from hematopoietic stem cells basically don't have cilia, as far as anyone is aware. So this must be some form of hedgehog signaling that doesn't require cilia, although these cells have can undergo a process where the mother centriole docks to the plasma membrane, which is one of the first steps in cilia formation, but somehow in immune cells that doesn't lead to a full cilium forming. But there are actually some similarities at the cell biological and even molecular level between some of the events that immune cells do and what ciliated cells do. So there may be some connection that remains to be fleshed out.
Last question.
Are there any actual structural differences between the plasma membrane that surrounds the cilia versus the
That's a great question. So we don't know. The short answer is probably yes, but in detail, we don't know all of those differences. So somehow, even though these membranes are in theory continuous, and that a protein sitting in the plasma membrane could diffuse into the ciliary membrane and vice versa, that doesn't actually occur in reality. So there appears to be some type of diffusion barrier that allows these two membranes to have different contents. Exactly how it works is not known. And likely the lipid composition and membrane composition of the plasma membrane and ciliary membrane are distinct in some ways that we know about, but probably in additional ways we don't yet know about.
Well, David, thank you for an excellent talk.
Thank you. Yeah. Thanks.